Hello and welcome everyone to this session of Bakul Talks about the Nobel Prizes of 2020. I'm sitting here at the Bakul Library, and at the library we used to organize many such sessions, meet the authors and experts talking about different topics. But then, with the pandemic, the library closed, all our activities closed, and then we kind of reinvented ourselves digitally. So we started with storytelling programs which are happening every Saturday at 9 p.m. We travel to different countries with one of the finest storytellers from that country. So those of you who are not aware of that, you could take note. Every Saturday at 9 p.m. we go live on Facebook, YouTube, and Twitter on the Bakul Foundation handles, where we travel to different countries. And every Sunday we have storytelling in Odia, which we call Rabibar Gopabar. So if you are interested in listening to stories, you can tune in Saturdays and Sundays at 9 p.m. live on the Bakul Foundation social media handles. And then we started Bakul Talks. The main reason behind Bakul Talks was, can we have public lectures that are of common interest to everyone? Because one of the problems that we realize is that now I'm a student of literature. I often feel like Penny, if you've seen, that was Penny in the intro video from The Big Bang Theory, the popular web series. And I often feel like that, what are they talking about? What are these big researches? I was a student of literature, though I studied science in school like everyone. And I often did not really make the effort to try and understand. Now, when it came to the Nobel Prizes, and when we talked to our young volunteers, everyone at Bakul volunteers, and they said, yeah, we understand the significance. We know that this is a major work which has impacted mankind, but we don't think we can understand it. So that is where we thought of conceptualized Bakul Talks, which are for everyone. So if it is a Nobel Prize in chemistry that we are talking about today, this talk is not only for students of chemistry. In fact, the idea is it is people from any disciplinary background, any curious individual, and we want to foster curiosity. In the session on the Physics Nobel Prize, Professor L.P. Singh talked a lot about the importance of fostering curiosity in people, and that is something that we would want to, and that is why we hope that there are many people here who are not really students of chemistry, but are curious to know about this groundbreaking research. So that is, and we have been getting the best of speakers in the country for this series. We have been lucky, and for example, today we are very happy that we have Professor Jyotirmayee Dash with us, who's a very eminent professor of chemical sciences. Now, this year's Nobel Prize in Chemistry is very special. It has gone to two women for the first time. In fact, in the entire history of 120 years, the Nobel Prizes started in 1901. And in 120 years before this year, only 5 women had got the Nobel Prize in Chemistry. That is the kind of underrepresentation of women scientists and their work, and this year's the awardees, 2 of them got it, and we are happy that Jyotirmayee Dash, who's a very eminent professor of chemistry, as I said, she also is a recipient of the Bhatnagar Award. Earlier this year, the Bhatnagar Award is popularly called the Indian Nobel Prize. It's the highest award given by the government of India for contribution to scientific research. And she is the first woman from Orissa, though I don't think we should look at her, in a narrow sense as from Orissa, because she is a scientist and she's contributing to knowledge in the world. But as someone from Orissa, definitely we are very proud of her. And not only that, she has also a strong connection with other Nobel laureates. For example, in the discussion on the Physics Nobel Prize, we talked about C V Raman's association with the Indian Association for the Cultivation of Science in Calcutta. And Professor Singh talked about the kind of accident that happened. How C V Raman had been employed in Calcutta actually as an auditor general, and in the auditor general's office, and he was passing in the rickshaw and he saw this building. It was the first research institution in the country for science. And then he went in there, and that is where his career in scientific research started. After, of course, his whatever he had done in school, college. And Professor Jyotirmayee Dash is a professor there. Not only that, She was also the Marie Curie Fellow at the University of Cambridge. So she has had an association with, in her life with Nobel laureates. She knows what it is to get a prestigious award in science, and she will be talking today about the very significant research. I'm told that the research of Emmanuel Charpentier and Jennifer Doudna is one of the most significant, which has really impacted, mankind. Just imagine if you can change the DNA if there is possibilities of curing inherited diseases. Now we are talking about using their work, the discovery of the genetic scissors, particularly the CRISPR genetic scissors, for also research on COVID. So this is something which has had huge impact on the life sciences. And today, Professor Jyotirmayee Dash is going to talk about that. I won't take more time. We'll go straight to her. But just before that, just one small announcement. You'll be very happy to know that we're starting a new program on Thursday called Travels and Novels. When we did this storytelling from around the world every Saturday, the Ministry of External Affairs of the Government of India took notice and they found it very fascinating. And day after tomorrow, actually, Prime Minister Narendra Modi is having a bilateral meeting with the Prime Minister of Luxembourg. And this is the first time it's happening in 20 years that the two prime ministers are meeting. And so the ambassador of Luxembourg reached out to Bakul and requested us to have a storytelling from Luxembourg. But this storytelling is not for small children. So we have a very famous author from Luxembourg, Anna Leader. She will be speaking about literature, about her writing, and she will be giving her perspective on a writer's perspective on the culture of Luxembourg. So if you are interested, please join us on Thursday at 9 p.m. on Facebook, Bakul Foundation handle on Facebook, Twitter, and YouTube. So that is the announcement for Thursday. Right now, over to Professor Jyotirmayee Dash and her talk on the Nobel Prize in Chemistry for 2020.
Can you just give me 1 minute? One second, please. Yeah, sure. Just a second, please.
Yeah. And a very interesting thing. I'm sitting in the Bakul library here in Bhubaneswar, and we showed many books on physics that are there in the library. When the library opens, you can come down. There are very interesting books. Can I have myself full screen, please? So that we can just see the book. Yeah. So this is a very famous classic book by Matt Ridley on genome. And there are other fascinating books that you can check out at the library also. And not only that, there are many other books. So you're welcome to come down to the library when the library opens, probably in a month's time, and check out these books. But now over to Professor Jyotirmayee Dash.
Yeah, good evening, everyone. At the beginning, I would like to thank for the invitation to deliver a talk. And this has been an excellent webinar series for discussing Nobel Prizes of 2020. So I am going to talk about the work that led to the Nobel Prize in Chemistry this year. So let us know, as this has been a webinar series on Nobel Prizes, so you know what is Nobel Prize and all. So before just I come to the today's topic on Nobel Prize in Chemistry 2020, so let us talk about a bit about the Nobel Prize. So it is the most prestigious award, and it is awarded to people whose work has contributed significantly to the benefit to the mankind. So that means their work is recognized because it has the ability, so it has the potential to benefit to the mankind. So the Nobel Prizes are awarded in 6 disciplines: physics, chemistry, physiology or medicine, literature, and peace. And this this prize is awarded after the name of Alfred Nobel, a Swedish chemist, engineer, and industrialist. So this is awarded as, he is from Sweden. So this is awarded by the Royal Swedish Academy of Sciences at Stockholm, Sweden. So now let us see about Nobel Prizes in chemistry, like down the years, like it started from, as he stated that it starts from 1901 and to the 2020. So this prize is awarded to the person who has made the most important chemical discovery. So, like Nobel Prize is given to the people whose work has contributed, whose work has immensely contributed to the benefit to the mankind. Similarly, it is awarded to the person who has made the most important chemical discovery, and that has also the impact on society, like whether it is health, environment, or industry. So this is so far, like it has been awarded 112 times to 186 Nobel laureates between 901 to 2020. So Frederick Sanger is the only Nobel laureate who has been awarded the Nobel Prize in Chemistry twice. And this prize covers or recognizes the whole sphere of the basic chemical sciences, from theoretical chemistry to biochemistry and applied chemistry. So at the end of my talk, after discussing about the Nobel Prize in Chemistry 2020, I'll discuss about the Nobel Prizes in different disciplines of chemical sciences. At least I'll give a few specific examples. So this year, the Nobel Prize in Chemistry has been awarded to Professor Jennifer Doudna and Professor Emmanuel Charpentier. So they received the Nobel Prize due to the development of a method for genome editing. So before we go to the topic, so let us see, like, sure, you see, this has been an exciting year also, as mentioned, in the introduction, that this year has been an exciting year for women scientists, like scientists who are women. There is no term like women scientists. So they have won the prize this year, and let us see, let us pay a tribute and see the other 5 women who have received Nobel Prizes in chemistry. Marie Curie was the first pioneering woman to win the Nobel Prize both in physics and chemistry, that is twice in a lifetime. And this has been a rare feat for any for anyone who has received a Nobel Prize in chemistry. So this has been a rare feat for, and that has been accomplished by 3 others since then. And then, you see the year 1911 for the Nobel Prize in chemistry. Then after 3 decades, it is her daughter, like Dr. Irene Curie, who won the Nobel Prize for chemistry. And then again, after 3 decades, it is Dr. Dorothy Crowfoot Hodgkin, who won the Nobel Prize in the year 1964 for protein crystallography. So the biomacromolecules like protein also been awarded in the area of chemical sciences. And again, after Dorothy Hodgkin, it is like after 45 years, it was Dr. Ada E. Yona who won the Nobel Prize for the structure of ribosome. And again, you see the trend is getting better. In the year 2018, Dr. Frances Arnold has received the Nobel Prize for the directed evolution of enzymes. So as we can see, 2009 to 2018, and this year, so the trend is, I can say, is getting better. So now let us know a few words about the Nobel laureates. Dr. Jennifer Doudna. So she is an American biochemist. She is a professor of biochemistry and molecular biology at the University of California, Berkeley. And she, before winning the Nobel Prize in chemistry, she has received several other prestigious awards for chemistry and biological chemistry and medicine. And Dr. Emmanuel Charpentier is a French researcher in microbiology, genetics, and biochemistry. She has also founded an independent research institute, and she is a professor and director of Max Planck Institute of Infection Biology in Berlin. So before again receiving the Nobel Prize in Chemistry, like Doudna, she has received many other prestigious awards. So now, so as in the first slide, I have shown that they have received the prize for a method for genome editing. So what is genome? Let us know, as few students, also school students are attending. So let us know some related terms. What is genome? So all organisms are made of cells. With few exceptions, all cells contain DNA except a few viruses that contain RNA. So genome is the total genetic material, that is DNA of an organism or RNA in viruses is known as the genome. So see, this is a typical cell. It contains the nucleus, and this is the chromosome, and this is the DNA. You see, the DNA duplex DNA is also, we can see, as a ladder. So we'll go a bit about the DNA structure and gene. So the DNA is the chemical code for making and also sustaining the life. You see, this is the double helical DNA structure which is found in any cells. So DNA is found in all living things, and it carries the instructions to make proteins. And you saw this is a chromosome. So this is are the coiled up strands of DNA is a chromosome, and the chromosome actually holds the information to make different proteins. And what is the gene? So genes are pieces of DNA that hold the information to make proteins. So there are a lot of DNA, as you are seeing, that chromosome is the coiled up strands of DNA. So, however, only less than 2% of our DNA contain genes, and that make proteins. And a chromosome has many genes. So now let us know the central dogma of molecular biology. So that is DNA-based gene expression, because when we're going to know about the today's talk on Nobel Prize in chemistry, so there DNA makes RNA and RNA makes protein. So this is the central dogma of molecular biology, or we can also say central dogma of the gene expression. That is the flow of genetic information from DNA to RNA to proteins in the cell. And now, so how do cells make proteins? So this is a process when DNA makes RNA is called transcription, and the RNA then comes out of the nucleus. You see, DNA is present in the nucleus, RNA is present in the nucleus. So therefore, the name is nucleic acids, that it is present in nucleus. So it is deoxynucleic acid and ribonucleic acid. So they can be translated to give proteins. So RNA comes out of the nucleus in the cytoplasm and makes the proteins. So now, so how the structure of RNA is different that of DNA. So the DNA and RNA both contain sugars and different sugars, so the name is different. So in DNA, it is deoxyribonucleic acid, it contains a deoxyribose sugar, and in RNA, it is a ribose. So therefore, the name is deoxyribonucleic acid for DNA. So there is no hydroxyl group is present here, and in the ribo, this is ribonucleic acid. All DNA and RNA contains a sugar, contains the phosphate group, that's why it is acid, and also contains the nitrogen base. And second is the bases. So both DNA and RNA contains bases. The DNA contains like So this is your RNA and this is your DNA. DNA contains 4 bases. RNA also contains the 4 bases. Cytosine is present in both DNA and RNA. Guanine is present both in DNA and RNA. Adenine is present also both in DNA and RNA. And thymine is present in double helical DNA, and the uracil is present in the RNA. So this is the difference, that uracil is present in the RNA and thymine is present in the DNA, but the structurally they are same. Only one methyl substituent is present in the thymine, is extra in the thymine of DNA. So those, as you see in DNA, 2 strands you are seeing, and they are held together by hydrogen bonding. So here adenine hydrogen bonds in thymine in DNA. Since thymine is absent in RNA and uracil is present, so in RNA, adenine hydrogen bonds with uracil, and guanine hydrogen bonds with cytosine in both DNA and RNA. So this is the basic and common structural difference. You see, the structure of Nobel Prize, structure of this double helical DNA was determined by Watson and Crick. So Watson and Crick proposed the structure of double helical DNA in the year 1962. Sorry, this is a name. This should be, a capital letter of Crick. So they won the Nobel Prize in Physiology and Medicine for proposing the double helical structure of the DNA. And now if you see, so this is like your and this is a phosphodiester bond, that is this one, as you are seeing the phosphate. The guanine hydrogen bonds with cytosine and adenine hydrogen bonds with thymine, and this is how the 2 strands are held together in a double helical DNA. So we know now what is gene, what is DNA, what is RNA, and DNA gives proteins. First DNA makes RNA, and then RNA makes protein. And let us know now what is genome editing. So genome editing is a technique of inserting, replacing, or removing, and even modifying DNA in a genome. So that means we can use the method, we can insert a piece of DNA, we can replace a piece of DNA, we can remove or also modify a piece of DNA in the genome. And it is mostly carried out using artificially engineered nucleases. So the nucleases, what is nucleases? Again, a new term. So nucleases are enzymes. So these are called molecular or genetic scissors. So nucleus is enzymes that cut the nucleic acid, DNA or RNA, and they create a single or a double strand break. So if it is a duplex DNA, so you see an example of a duplex DNA. As you know now, these letters are present in the double helical DNA, like G, C, A, T, and G will hydrogen bond with C. So the complementary strand will be the one where the nucleobase pairs and does the base pairing, like guanosine hydrogen bonds with cytosine. So in the complementary strand, you have cytosine, and guanosine. If it is thymine, then we have an adenine. So this is your nuclease enzyme. So it can cut this duplex DNA and we can say that it can cleave the DNA and it can create a double strand break. So now you see an example. So this is the recognition site. I mean, this is where the nucleus cleaves the double helical DNA. So it cleaves the phosphodiester bonds, and we can get this. So this is the phosphodiester bonds. So if you put a nucleus enzyme, so it cleaves. Enzyme means it's also a protein, and this protein is also made by DNA, that is your gene. So now let us know what are the genome editing methods. The CRISPR-Cas9 is the recent method. So this is not the only one method known for editing genome. So genome editing mainly consists of 2 phases. The first is to create a double-strand break by using genetic scissors. So you see this is a duplex DNA. So you're seeing a cleavage, that is a break. So this can be done so there are different nuclease enzymes. One is meganucleases. The second one is zinc finger nucleases. talents, that is transcriptional activator like effector nucleases, and the most recent one and the most efficient one is the CRISPR-Cas9 genetic system. So now the second, once these nucleases create a double strand break, cleave the DNA, then the cell uses its own repair mechanisms and fixes it. Sure. So this can be done in two methods like non-homologous end joining and homologous directed repair. So these are the two different methods. One is homology directed repair and another is non-homologous end joining. So your seizure, what it is doing, your genetic seizure, or you can say molecular seizure, cleaves the DNA and make a double strand break. And then the cell allows its own repair mechanisms to fix it. So if we can insert, so when the cell is joining itself, so here in case of non-homologous end joining, so random DNA is inserted or deleted. So this would result in knocking out of a gene, so a gene is deactivated. So by editing DNA, we can knock out of a gene. So we can remove or we can deactivate, for instance, a cancer-causing gene. And another is a homology directed repair. So in this case, we can insert a donor DNA, and here we can replace a gene. And this method can activate a new gene. So now, so what we have seen so far, that what I have shown, that genome editing method, there are 4 different nucleases. One is meganucleases, zinc finger nucleases, TALEN, and the CRISPR-Cas9 system. So all these nucleases can create a double strand break, so can cleave the DNA, can cut the DNA into 2 pieces, and then so there is the repair mechanisms. We can insert a new DNA template and we can repair in 2 different ways. So the cell can repair in 2 different ways. So this is called genome editing. So now let us see. So in the previous applications, like in the previous developed molecular seizures, like meganucleases, zinc finger nucleases, and talents, there are several limitations like low efficiency, the process is time consuming and laborious, and they can only target only a single site at a time. But the CRISPR-Cas9 system gaining attention since 2013. So it has received considerable attention since 2013, and this is a RNA directed nuclease system. So let us see. So CRISPR, since it is developed from 2013, there is an unprecedented rise of CRISPR. You see, the yellow ones are CRISPR. So there are a lot of publications, whether it is in the title or abstract, you can see. So this has been significantly increased. So this has been significantly used for genome editing in comparison to other reported or known methods. So now let us know what is CRISPR-Cas. So what is the CRISPR stands for clustered, regularly interspaced, short palindromic repeat. So clustered, regularly interspaced, short palindromic repeat. And Cas is CRISPR associated protein. So CRISPR, you say this is a bacterial chromosome. So in this bacterial chromosome, so the name would be simple if you see this. So in this bacterial chromosome, we are seeing like this is a CRISPR locus. CRISPR locus means you see the CRISPR full form, that is, it contains clustered DNA repeats. So these are, you see the hexagons, black hexagons. So you are seeing this is repeated 1, 2, 3, 4, 5. So this is regularly repeated, and this is also separated by shapers, by spacer. So by, you see the colorful spacers, blue, yellow, and green. So what are these spacers, and what is this? All are DNA. So this is so they are separated by spacers. So these spacers, so the name comes that interspaced. So this is the clustered regularly repeats, and these are short repeats. That is not very long DNA. but it has short DNA repeats, and only one term remain, and these are interspaced. So you now understood that it is clustered. These are regularly interspaced, and these are short, and these are repeats. And only one thing is what is palindromic. They should be also palindromic. So let us see what is palindromic. Palindrome means a word when we read from the opposite is the same when we read from the front. So let us have a few examples. Mom. So if we read from this direction forward or backside, so we can read as mom. Madam, another word. Malayalam. So if you read from this direction or the opposite direction, so they are same. So now let us see an example of a double-stranded DNA sequence that is palindromic in nature. So this is one example. So this is, you see, TTAGCAC, and this is from forward direction, and you see from the opposite direction the same two T's, A, G, C, A, and C. So this is a palindromic DNA sequence. So those black hexagons, what you were saying, are palindromic in nature. So now let us know that CRISPR-Cas9 system, this is a bacterial immune system that protects them from virus. So let us know a few terms, and first let me say you something like bacteriophage. Bacteriophage is a virus that attacks a bacteria. So viruses that attack bacteria are called bacteriophages. So bacteria have their own defense mechanism to destroy the virus. So when we are attacked by the pathogens, when we are attacked by the pathogenic organism, for instance, virus, so our body's defense system tries to nullify it. So they try to nullify the effect of pathogens, and sometimes they win, and sometimes we also win. So similarly, when the viruses attack the bacteria, sometimes the virus wins, and sometimes the bacteria wins. So let us see CRISPR-Cas with one such defense system, or we can say immune system of bacteria that protects them from the virus. You see here, this is a bacteriophage. So they have their viral DNA. So a virus contains either RNA or DNA as its genetic material. So here, that is in the bacteriophage, it contains viral DNA as its genetic material. So it sits on a bacterial cell and injects its genome, that injects its DNA into the cell. So now the viral genome takes hold of the bacterial cellular machinery. So what happens, that viruses, which are known as bacteriophage, so they attacks a bacterial cell, and injects their viral DNA into the bacterial cell. So before going, let us know some of the related terms. I said you bacteria, I said you virus. So bacteria are microscopic. You say these are bacteria, single-celled organism. They have only single cell, and that thrive. That is, they try to survive in diverse environments. So they live in soil, in ocean, and inside the human gut. And humans' relationship with bacteria is quite complex. Like, they are mostly harmful to human beings. Sometimes also bacteria lend us a helping hand. For instance, curd. The bacteria present in the curd helps with our digestion. So now bacteriophage, you saw this type of bacteriophage was sitting on the top of a bacteria. So a bacteriophage is a type of virus that infects bacteria. So bacteriophage can destroy their host bacterial cells, and all bacteriophages are composed of nucleic acid, that DNA or RNA molecule, and that is surrounded by a protein structure called capsid, though we don't need to know capsid for this work. So now let us see how virus infects the bacteria and kills the bacteria. So this is, you have already seen that bacteriophage sits on the host bacterial cell. So this is the foreign DNA. So it is injecting its DNA, this green color DNA. This is the phage DNA. We can say this is the foreign DNA, which is entering the host cell, that is your bacterial cell. And I have just shown that is a capsid, that is the protein which is present in the bacteria. So first step is attachment of the phage to the surface of the host. So like it is, it sits on the top of the bacteria, or it attaches itself to the surface of the host bacteria, and then it penetrates its viral DNA into the cells. So then the third step is biosynthesis. Like fast DNA replicates, DNA, I have in the first slide I have shown you that central dogma that DNA gives RNA and gives proteins. So fast DNA starts replicates and fast proteins are made. Then the new phage particles are assembled. So it's good for the virus that it uses the bacterial cell to replicate, and then the number of phage particles, then the cell ruptures and the bacteria is killed. So now let us see how the bacterial adaptive immune system protects bacteria. Now, certain bacterial species, they have CRISPR-Cas genetic system. So that helps the bacteria to degrade the viral genome. So let us see how does this help the bacteria to degrade the viral genome. Like it helps the bacteria to degrade the viral genome. Not only that, it also affects the future attacks by the same virus. So the virus cannot attack again. If it attacks, it will be again degraded. So let us see this process step by step to understand what is CRISPR-Cas. So this is the CRISPR-Cas system, which is called adaptive immunity in bacteria. So this is your bacterial cell, and this is the phage, your bacteriophage. So it sits on the cell. So it involves number of steps. So three stages. So I have shown here three stages. Like stage one, foreign DNA acquisition. That is your phage penetrates its DNA. And you see, this is the CRISPR locus. I'll come to it again. And then second stage is CRISPR RNA processing. That is CRISPR DNA gives CRISPR RNA. Then this is a long RNA. So this is cleaved and you get mature RNAs. So that then forms RNA guided targeting of the viral elements. So the three stages now will go step by step. So in the first step is the bacteriophage penetrates its DNA into the bacterial cell. So now you see this is the CRISPR locus. You know that is repeats. You see here now it is represented as black diamond units. So black diamond blocks. So they are repeated, and they are also, so there is also spacer. You see green, so red, violet, yellow, and green. So these are repeats. So these are viral DNA from your bacteriophage. So now let us go to the next slide. So what you see, so I just took out this part, the Cas genes and the CRISPR locus. So certain bacteria, have their this CRISPR locus, that is the palindromic DNA sequences. So what is it? So you see the black diamond blocks are the palindromic bacterial DNA sequences, and which are repetitive in nature. For instance, if it is TTA-GCSE, so in each of these blocks would contain this TTA-GCSE. And the colorful blocks that we are seeing, red, violet, yellow, and green, are part of different viral DNA which are incorporated within the CRISPR locus of the host bacteria. That means this DNA, this part of the DNA contains both bacterial DNA as well as the viral DNA. And so this is called CRISPR locus. That is an array of palindromic DNA repeats, the black diamond bugs, that are interspaced by the parts of the different viral DNAs. So now let us go and for this, as you are seeing, the DNA is coming and connected, the viral DNA is connected to the bacterial DNA. So that means it is done by different enzymes. So we are not going that detail. We are just knowing that there are different enzymes like Cas1, Cas2 complex that helps in the insertion of the viral DNA fragment into the host bacterial genome. DNA polymerase, so they helps in the synthesis of new DNA repeat sequences after the viral DNA incorporation, because DNA is incorporated and this is like interspaced and in a repeated manner, and DNA ligase, so this would connect two pieces of DNA together. So now we have, this is your bacterial chromosome, and this is your host genome. Host means your bacterial genome, and this is your foreign DNA in the bacterial genome, that is the viral uh, DNA, that is the phage DNA. So now, as you know that this, I have shown the central dogma that DNA gives RNA and RNA gives protein. So now this DNA, that is the viral DNA plus bacterial DNA, will transcribe. The process is transcription. So by the transcription, the DNA will make the pre CRRNA. So what is CRRNA? That is pre-CRISPR RNA. So this is a long primary transcript. And they contain, as you see this structure, these are RNA. These are not double helical DNA as it is present here. These are single stranded RNA. But if you can see, this is like hairpin structure. So here there will be like double helical structure, and this is like a hairpin. So this is called pre-CRRNA. So it's a content of series of RNA hairpins, which are also called RNA secondary structures. During the meantime, as you were seeing, it contains Cas genes, and since this DNA gives the RNA by transcription, these Cas genes will also, since these are genes, they will convert to the corresponding proteins. So during the same time, the Cas genes will give Cas mRNA by transcription, and then Cas RNA will give Cas9 enzymes. So in your bacterial locus, you have CRISPR RNA and you have now Cas9 enzyme. So now this is, so you are seeing, again, I'm just going to explain this, that these are bluish blocks. So these are different Cas protein encoding genes. So there are several Cas proteins like Cas3, Cas6C, Cas6F, Cas9. So Cas9 protein is present in Streptococcus pyogenes, a bacterial species. So this is Cas9 protein was identified by Charpentier's group, and they have worked in these bacterial species, that is Streptococcus pyogenes. So now next, what happens, as you see, that this CRISPR locus, CRISPR DNA gives your CRISPR RNA, and then you are seeing these blue positions are cut, like this RNA is also cut by an enzyme. This is, you can say, also molecular seizure. This is also a nucleus enzyme, but this is RNA specific. This is RNase. So the long pre-cRNA is then processed into smaller units. That means every spacer, you see red spacer is present here, and this black diamond is black RNA. So this is from this black part of RNA molecule is from your viral RNA, and this red RNA is from your, so the black one is your bacterial RNA, and this red one is your viral RNA. Similarly, this black one is from your bacterial RNA, and this green one is from your this viral RNA. So that means your CRRNA is now, so this is now, you can say, CRISPR RNA. So this is now processed and matured, and these are smaller units, and they all contain a single spacer to a black hairpin. So a hairpin and a single spacer. The spacer is from viral unit, and the hairpin is from your bacterial unit. So the CRRNA processing is also dependent on bacterial nucleus enzyme, so which are selective for RNA, which I've already mentioned, and this is the RNase. So now this matured CRRNA after the processing step would form a complex, that is CRRNA Cas9, that is ribonucleoprotein complex. So this is like RNA guided targeting of the foreign and viral DNA. Sure, you see the process is not that simple. So it is like a programmed manner. You'd be surprised how the nature makes this system so easy, that like bacteria has its CRISPR-Cas9 system to kill and destroy the virus. So now the CRISPR RNA, so this is your CRISPR RNA that we have seen, that black hairpin. and the red special. So now it is shown in the blue color. So this is the CRISPR RNA. So this will now recognize the new DNA that is coming from the virus. So since the CRISPR RNA part contains the viral RNA itself, so it will recognize the DNA part by complementary base pairing, because it's its own RNA. So it recognizes by complementary base pairing. Now the CRISPR RNA is connected to the Cas9. So this is your Cas9 enzyme. This is also quite complex enzyme. It is just shown in a simple manner. So this is also connected to the Cas9 by another RNA. So this is like sort of programming. Another, you see, tracer RNA is also present in the bacterial genome. So this is fixing it and making also a complementary base pairing. So now, so this is called the tracer RNA, and this is your CRISPR RNA. So this combination of CRISPR RNA and tracer RNA is known as guide RNA, because they base pair and make one RNA. So that we can say as a guide RNA. So now the CRISPR RNA will only recognize the viral DNA only if there is a PAM sequence is necessary. So the PAM sequence is necessary for the viral genome for creating a double strand break. So for creating a double strand break for the Cas9 and CRISPR RNA, so we need a PAM sequence here. So this is what is a PAM sequence. So this is a PAM is called protospacer adjacent motif. So this is adjacent to the point where the enzyme would cut the DNA and would create a double strand break in the DNA of the viral DNA. So here, what I said, this is usually 2 to 6 base pairs in length and mostly found in the 3 to 4 nucleotides, as you know, ATGC like that. So for S pyogenes, it is either 5 prime NAG. So NAG means adenine, guanine, any nucleotide. So this is if AG is present, then then the Cas9 and CRISPR system can cut this DNA and create a double strand break. Or NGG, like 2 guanosine motif and any other nucleotide. N stands for any other nucleotide. So the PAB motif is important. Why it is important? Because it helps the host bacteria, because bacteria is recognizing the DNA of the viral DNA, that recognizes only the viral DNA and spare its own DNA. Otherwise, it will go and bind to own DNA and cleave it. So it won't happen. So thus, any self bacterial DNA is not degraded. So because of the PAM is required, and PAM is only present in the viral DNA. So this is quite selective to cleave the viral RNA and not the bacterial RNA. So therefore, any cell bacterial DNA is not degraded. Only viral DNA which gets injected from the outside is targeted and degraded. So now you see this is your CRISPR-Cas system. The viral DNA is recognized by the guide RNA. Guide RNA is combination of the CRISPR RNA and tracer RNA, and Cas9 nucleus cuts both the strands of the double-stranded viral DNA, creating a double-strand break. The degradation of viral DNA is performed by specific Cas nucleus like Cas9, and this complex, now you see this complex of Cas9 and CRISPR RNA remains ever ready within the bacteria. So this system is ready within the bacteria to hunt down and degrade any viral DNA it enters. So since this is ready, so whenever any particle attacks or something, it will go and cleave their DNA. So first CRISPR-Cas9 cleave the RNA, cleave the viral DNA, and further it has its own complex that it can also degrade any viral DNA enters the bacterial genome. So now let us watch a short video on the CRISPR-Cas system. Okay, so now let us summarize the CRISPR-Cas system. The CRISPR-Cas system consists of key molecules that introduce a change into the DNA. So these are an enzyme called Cas9. This acts as a pair of molecular scissors that can cut the two strands of DNA at specific location in the genome. A small piece of RNA called CRISPR RNA, which is about 20 bases long, and which has been transcribed from the viral and bacterial DNA parts. So this is your CRISPR array. And CRISPR RNA combines with the tracer RNA to form the guide RNA, and then the guide RNA forms a complex with the Cas9 enzyme. PAM sequence in the viral genome is necessary for the recognition of the viral genome and its cleavage. This then targets the viral DNA, and Cas9 enzyme cuts at the right point in the invading, that is your infecting viral genome. So now let us know how the CRISPR-Cas9 system, which is a natural bacterial immune system, has become a gene editing tool. So now this is the timeline of CRISPR-Cas9. So CRISPR repeats was first observed in bacterial species in 1987. So it was by a Japanese scientist from the Osaka University. And in the year 2002, the CRISPR elements, that is, and associated genes like Cas genes and also CRISPR elements, that is, this is your CRISPR here. So these repeats, DNA repeats are present. So this was identified in the year 2002. And in the year 2005, CRISPR spacer was identified as a part of foreign DNA. So this is your viral DNA, that is your foreign DNA. So this was identified in the year 2005. In the year 2006, CRISPR proposed to be a bacterial adaptive immune system. So this was known before. In the year 2007, discovery that CRISPR-Cas imparts resistance to specific phages, like bacterial resistance. Sometimes they develop resistance to certain things. Similarly, CRISPR-Cas also becomes resistant to specific phages, so they cannot destroy certain phages. Then in the year 2010, CRISPR-Cas was identified as a bacterial and also an archaeal immune system. So this is the archaea. This is also a single-celled microorganisms mostly found in the water and all. And in the year 2012, so this is where the work of Charpentier and Darna, otherwise CRISPR-Cas system, was known as a bacterial adaptive immune system. CRISPR-Cas9, the Cas9 protein, was identified by the Nobel laureates, and this was also developed as a gene editing tool. So this was done by the Nobel laureates. So now let us see a bit. So then in the year 2013, CRISPR-Cas9 used to edit targeted genes in both human and mouse cells. So the method in the year 2012, what was developed by Charpentier and Darna, was used to edit targeted genes in both human and mouse cells. And then CRISPR-Cas9 system was also used in plants to modify crops. And in the year 2014, monkeys with CRISPR engineered targeted mutations are born. So, and in the year 2015, it was also used to develop virus-resistant tomato plants. And in the year 2016, that is your US Department of Agriculture determines CRISPR-Cas9 crops will not be regulated as your genetically modified organism. And in the year 2017, US Patent Office awarded key patents to the CRISPR-Cas9 based. Several patents were submitted, and the key patents were granted in the year 2017. And in the year 2020, the CRISPR genome editing got the Nobel Prize. So now let us see what they have done. So in the year 2012, like Emmanuel Charpentier studied Streptococcus pyogenes, which is harmful to the humanity, and her work showed that tracer RNA is a part of bacterial CRISPR-Cas. So that can disarm the virus by cleaving their DNA. And in the year 2012, Dr. Charpentier initiated a collaboration with Jennifer Doudna, which is an experienced biochemist with a vast knowledge of RNA. And together, they have succeeded in creating bacteria's genetic seizures in a test tube. And then they have simplified the seizures molecular components so that they can be easier to use. And in the 2013, they have demonstrated that Cas9 CRISPR can be used as an engineering tool in eukaryotic cells. So this is the paper they have published in the year 2012. So this is a programmable dual RNA-guided DNA endonuclease in the adaptive bacterial immunity. This is by Doudna and Charpentier. So in this case, what is required is a dual RNA. That is, we need both CRISPR RNA and tracer RNA for the binding and cleaving the target DNA, requirement of PAM, and then it was found out that Cas9 can be programmed with this single, chimeric RNA, and Serpentier's group has identified Cas9 protein in the Streptococcus pyogenes. Sorry, is the time is up? Time is up.
No, we'll just close it soon because I think on Instagram it will end in 2 minutes. But we'll continue on YouTube and Facebook. So
okay,
yeah,
okay,
you can go back and we'll try and finish the talk so that we can have some time for questions.
Okay, in 5-10 minutes, but I had a lot of things. Okay, so maybe a little bit I'll just so and go to the last. Sure. Now. So this is the CRISPR-Cas9 system, which was developed in the Streptococcus pyogenes by Jennifer Doudna and Charpentier. And this is the Cas9 protein, which is quite complex. And this is like an RNA-guided DNA cutter. So this can cut the RNA. So now let us see quickly that application of this CRISPR-Cas9 system from an immune system to an engineered technique. You see that we have seen that 2 RNAs are required in the natural system, but a guide RNA can also be engineered. Like what we can do that the target RNA can be, this is the complementary target RNA can be added to the tracer RNA. So although what we have seen that nature uses tracer RNA and CRISPR RNA, a single RNA guided Cas9 can be programmed for DNA cleavage and genome editing. So therefore, CRISPR RNA can be combined with the tracer RNA to engineer guide RNA, and the CRISPR RNA should be, this CRISPR RNA should be complementary to the target gene sequence that we need to edit. So this is the basic DNA editing using CRISPR-Cas9. So we need a guide RNA and we need a Cas9 protein that can be delivered into a cell, and then the cell can be edited. So that means the Cas9 gene from the bacteria can be transfected and expressed in any cells like plants and humans, and engineered guide RNA can be transfected, and Cas9 protein and RNA would now recognize the specific region of DNA and cut the DNA sequence. So what we can do, that we can add, remove, or alter a part of human DNA. So we can make specific codes or specific DNA sequence, and by editing the genome, we can change the characteristic of a cell or an organism. So by genome editing, we can edit a gene so that which in turn we can edit a trait. So we can change the trait, for instance, the color of the skin or the color of the hairs, and also gene for teeth development. So by editing the gene, we can do we can change a trait. So now let us see, CRISPR-Cas9 system has been used for disease treatment. So it has been used for cancer treatment, that we can delete the oncogenes by using CRISPR-Cas9 system. And if somebody is interested can see this excellent review article. And this system can also be used for HIV/AIDS treatment. Like similarly, like host gene, like this is the HIV virus which causes AIDS. So within the whole host cell, the cell RNA can be converted into the DNA, and with the help of reverse transcriptase. And once the DNA is formed, then the guide RNA would recognize the HIV DNA, and the Cas9 could create a double strand break and leading to the HIV DNA treatment. So thus, CRISPR-Cas9 system can be used for cancer treatment, for HIV/AIDS treatment, and it can also be useful for COVID treatment. But the method is same, only one has to engineer the specific RNA which is required to make a recognition of the corona virus. So further, the CRISPR-Cas system has been used to modify crops, like it can be used to modify corn, rice, sweet orange, tobacco, wheat, potato, and soybeans. And so these are taken from the talk from the Jennifer Doudna's talk, that is CRISPR opportunities and challenges. So it can be used for research, public health, agriculture, and biomedicine. And this is the future of CRISPR-Cas9. Like it can be used for genome engineering, like agriculture, crops, human gene therapy, and identification of specific drug targets, and also programmable RNA targeting. Another important thing is it can be used for public health, like suppression of the population of the mosquitoes, so that the mosquitoes won't spread. It can prevent the spread of mosquitoes. And another thing is CRISPR-Cas9 as an engineering tool, it can target multiple DNA sequences at once, and it has limitations, like it is required a PAM motif, and if in case there is a mismatch in the guide RNA sequence with the target gene, the guide RNA will base pair with another gene, leading to off target. So I think now I should stop here. Hello?
Yes, I think let us take some questions.
Yeah.
Can we exit the presentation? Yeah. Thank you so much, Professor Dash. I mean, it's a very detailed. I mean, I could feel, I left studying science so many years back, but I could actually, it was like from a beginner's guide to very complicated stuff you were trying to explain. I can understand that it's so difficult to explain to someone like me who does not follow science, starting from the very basics. But there was before we go back to the CRISPR genetic scissors, there was a very interesting thing she mentioned about Marie Curie and the other Nobel laureates, who happen to be women. As she said, very interesting. There's a difference between, calling women scientists and scientists who are women. And I remember reading this, that Marie Curie was the first woman to ever get a Nobel Prize. And in fact, initially, her name was not there for the Nobel Prize. And her husband, Pierre Curie, was there, and Bakul. And at that time, and it was the third year of the Nobel Prizes, 1903. And apparently he complained and he said that no, she has to be acknowledged. And of course, things have changed from then. But coming back to this, I'm sure all of you, thank you very much for staying on. I know that many others probably had to leave in some time, and on Instagram we ended. But then any questions you have for Professor Dash on the topic, or if you want to ask her anything about her own research, you can always ask. There are some questions which have already come in. So in your comments box, you can type in your questions now. Professor Dash, we have 10 minutes. You can be there for 10 more minutes, 10, 15 minutes.
Yes.
Take questions. Yeah. Okay. So we'll take questions now. And let me just remind you, if you're watching it on Facebook or YouTube, please do not forget to like and subscribe to our channels and to share this video. If you're liking this video, if you think that you learned a lot from this video, then please share this video so that others can also, get the benefit of it. So, and let's see what questions we have. Okay. So we have Swayamjit asking, he's a student of class 6. What is the use of replacing a DNA? Well, I think she already talked about it, but very briefly, if you can.
So if you can replace a DNA, like you can modify a DNA, you can change a characteristic, like you can change, deactivate a gene. For example, a gene is a cancer causing gene, it's an oncogene, so you can just replace that gene. So, and modify with a gene which is not a cancer causing gene or just a piece of DNA. So you can remove a part of gene which is causing a disease, and you can add a new gene if you want to try to see something like some changes like you see in the crops, like you can make them like a resistant free, virus free crops and all those things. So this is how you can change the complete characteristic if you replace the DNA.
So Darshana is asking, what is the scope of CRISPR-Cas in agricultural crops like sugarcane?
Yeah, sure. As I have shown a slide, quickly. Of course, I first, give a very detailed explanation of the CRISPR. I should have gone more towards the application. So this, CRISPR-Cas system can be used for the modification of agricultural crops. Like you can, have some mutations so that you can make a different colored things, or you can also make like tomato, like virus-free tomato and all. So there are many plants have been studied, and not that all plants have been studied. So it definitely, since it has been used for potato and tomato plants, it can be used for the sugarcane. So it has to be studied. It's not that all the plants and have been studied using this method. As you were saying, 2012 this paper was published, and within 8 years they received the Nobel Prize. So they have received the Nobel Prize. So it itself says that a lot of things to be done. A lot has been done, and lot has to be done.
So I think Balochan is asking cancer
No,
medicine.
no, I have seen the question. No, cancer medicine hasn't come. See, I have given a reference. You can see that is the review article of collection of articles which which have been published on the basis of use of CRISPR-Cas system to delete a cancer-causing gene or creating a mutation in the cancer-causing gene. So it hasn't come as a medicine. It rather as a gene replacement therapy to cure cancer.
And
So
that
this is
is
against studies.
and that has been effective in kind
This
of
has been studied in the in vitro models, in the cellular models, in the mouse models, and all.
okay.
So
So
it's in the
Safula,
research in the labs.
so Safula is asking, can the COVID virus pandemic also be in a way avoided by DNA editing? Is that possibility exist?
Definitely, that's what I was my proposed. I did not find much paper in this regard. Maybe people are working to use the CRISPR-Cas system to edit the gene of that coronavirus, that to kill the coronavirus and all. So that is also possible by
So
using
how
this
would it
method.
work? Because I did read somewhere when I was that that apparently there is some research going on in
Yeah, exactly.
on
Research is going on.
but
Definitely.
how would the genetic scissors help there?
So there, this is an RNA, corona is a RNA. So you can modify the nucleus so that it can recognize the RNA and clip the RNA. Or RNA can give the DNA, it can go and clip the DNA. So then the coronavirus gene, which is infecting and which is causing the disease, will be destroyed, will be clipped. The same method, the way bacteria is using and killing the virus, similarly, this method can be used specifically to kill the virus.
So I think there are tremendous possibilities. Everyone must be looking forward to
Yeah,
what
sure.
this will
Yeah, definitely.
Yeah. Okay. So let's see. Yeah, there is an interesting question that Sudiksha has asked. In my locality, I am getting to hear that the fruits and vegetables are hybrid. Are these the results of gene editing?
No, it may or may not be gene editing, but no, like as I have shown in one of my slides, like there are like these particularly the things which are modified by CRISPR-Cas9. So these can be allowed. This can be it has no problem. It can be allowed, and it's not hybrid. But definitely hybrid system or hybrid vegetables are coming to the market. But particularly this method has no effect. Somewhere now I have shown in one of my slides, like yeah, 1 second. the guidelines, that the timelines, that USDA, means US Department of Agriculture, determines CRISPR-Cas9 edited crops will not be regulated as GMOs, that is genetically modified organism, so it can be safely used.
Okay. So I think Shatabdi is asking, how long is it going to take us to study all the strands of DNA?
It's really a long process because people are seeing or modifying a DNA of their interest. So it's depend on the research. Like if you want to, there are oncogenes. When I say that cancer causing genes, there are several oncogenes. Like some genes are causing colon cancer, some are causing breast cancer, some genes are causing different types of cancer. So whoever is studying, they cannot do all the research work. Like they have to concentrate on a particular topic. So it takes a long time. So not that all the cancer causing genes or oncogenes have been knocked out by using this method.
So Jaga is asking, in which condition non-homologous joining and homologous directed repair are used?
Actually, 1 minute. Actually, I could not show all my slides today. You see, this is a repair mechanism of the cells. Since you are in BSc already, botany, so you can go through this one. This work by 1 second. I had a lot of slides to show you. That is, you see, the 2015 Nobel Prize. Can you see this slide? Or no, they cannot see anymore. You see, the 2015 Nobel Prize was given for the mechanistic studies of DNA repair. So they have received the Nobel Prize, like how the cells repair the damaged DNA. So if you look at the Nobel laureates like Linda Hall, Modris, and Sankar, then we can know a fundamental knowledge of how a living cell functions. So therefore, our genetic material all the time it's getting damaged and disintegrated, but it does not disintegrate. It does not disintegrate because a host of proteins, they work as a molecular repair kit. So therefore, if you go through their work, so you can understand about this that two conditions that I have shown you, like homology directed repair and also non-homologous repair. So this is cell has its own repair mechanism. So when it is a non-homologous enjoining, then you see just some mutations can be done and you can knock out a gene. But when it is homology directed, so you need a donor template to insert so that they will be similar. Now homologous means similar sequence as in your at the site of cleavage. So this, by using this, you can replace a gene so that you can activate a new gene. Okay. So this is, so you can go through their work as you are already BSc. I think this is also something you can study a little bit. So these are the two repair mechanisms that cell uses, and you have to also use the DNA donor template if we are working outside and studying the repair of the cells.
So this question is from Amrita. She's asking, DNA and RNA perform specifically if they're mismatched, so would become a harm to society. I think, yeah, there are of course concerns about gene editing and ethical questions also around that. So maybe you can talk a bit about that.
Yeah, I really didn't understand. So this is like DNA and RNA perform specifically if they are if they are mismatched. So sometimes if they are mismatched, then we don't get the result. It won't be any harmful. So whatever we are trying to do, maybe we cannot do. Like it would be off target. Like it is not going to target your target gene which you want to modify or edit, but it will go to somewhere else, or it may not go anywhere else. So these type of side effects are there for everything. If you take a medicine, there are side effects. So this is a common thing, that is possibility that if it is not no specific, but this system is very specific. As we are engineering the CRISPR RNA, that would go and specifically recognize the DNA of the particular target DNA that we want to modify or edit.
But the related question that I was talking about, I think I read somewhere that Jennifer Doudna, I think, has made a statement that she does not approve of gene editing when it comes to human beings. Because I think
that is your, because that was ethical guidelines, so that will not be done. Instead, you need to
so
do
that
something for medicine or something. Otherwise, that is like they are doing in the cells and seeing how it is working. And another thing also I want to say here, because I could not say you, as you see this year the Nobel Prize, they have received in the area of nucleic acid research particularly. So this is not the first time. That is what my slides, last slides were there, few slides were there on that there are several times the nucleic acid research has been bagged prizes in the chemistry. So this is not the first time. As I have shown you, DNA repair. So DNA repair 2015 Nobel Prize was also gone to the in the chemistry. And similarly, another thing is like you have seen that that viral DNA is connected with your bacterial DNA and giving you the CRISPR locus. So that is also a recombinant DNA technology. So that also received the Nobel Prize in the area of chemistry only, that nucleic acid chemistry. So those slides or few slides were there. I wanted to show you, but anyway, that was okay.
Yeah. So Abhijit is asking, he's in class 11, what are the possible requirements for replacing the DNA part? Also, is it helpful in any nuclear attack? Are there any other alternatives if it fails to cure?
Nuclear attack, I don't know if it would be really useful. So when you need, as you know, that when you are going to replace the DNA, you need your molecular scissors. And in other cases, that is just a nucleus enzyme, but here it is your RNA directed enzyme. You have a CRISPR plus Cas system. So that is what you need to replace your DNA. But I cannot comment on any nuclear attack. And really.
No problem. Yeah, I think he's just being curious.
Curious to know.
Yeah. So Jaga is asking, what is the role of track RNA?
So yeah, this is a tracer RNA. As I have shown you, this is used by the bacterial own natural system. So you can see here. You see, this is a tracer RNA. So this tracer RNA forms a complex. You see, this is also recognition by complementary base pairing, that RNA recognizing the DNA. So this is forms a guide RNA. So now the combination of the CRISPR RNA and tracer RNA is called a guide RNA. Now in the bacterial natural system, there are two RNA, like CRISPR RNA, which is coming of course from the your foreign DNA and transcribed to RNA, and the other is tracer RNA. But when it is engineered, that is also the CRISPR, that is the unsharpened work. So when that is done, you see, so that they have done in a single RNA. So there is not two RNA. So what is it? In the natural system, we have two RNAs. Like this one is tracer RNA, you see this stem loop structure. But here you can, since we are engineering, we can just put the complementary target RNA. So this is the part which is going and recognizing the viral DNA. So we can just make the primer, we can say a primer. So this sequence can be inserted, added to the tracer sequence, tracer RNA. So we can just make one guide RNA. We don't need, when we are engineering, we're making a tool because we are synthesizing and transfecting into the cells. We are adding it to the cells. So these are, so here we can take this guide RNA and can add to the cells. So this is called tracer RNA, but we don't need a tracer RNA separately. We can just use a single guide RNA for engineering, for genome editing. So that is what engineered. That is also Doudna's and Charpentier's work. Okay, I think it's clear.
Yeah. We'll just take the last question, and that is Balochan is asking, if a person has multimyeloma, can it be cured?
One has to use and see the CRISPR-Cas9 system. I don't think research has been done, but the thing is, it is now showing a promising Yeah, it is showing some interest, like it is showing some promise that it can cure cancers and it can replace the cancer causing genes can be replaced easily. So possibly, so that I cannot say. mm, Okay.
Sure. So thank you very much, Professor Dash. I think it has been enlightening. I think this is like I'm sure many people will keep going back to this video to, understand it better, because you've really covered a very wide canvas and like a beginner's guide into, and not only talked about the contribution of, the Nobel laureates of this year, but in the process of explaining their work, I think you've given a background and understanding to the entire topic. So thank you very much. And thank you all of you for listening to this. We'll be back, of course, next Tuesday with another Bakul Talks. And that time it will be next Tuesday. We're going to talk about the Nobel Prize in Literature. But as I said, you don't have to be a student of literature. And of course, literature is something that, of course, everyone reads and engages. And this year's Nobel laureate was the famous poet Louis Gluck, and we have a very famous poet in India. One of the best persons, again, you can think of Ranjit Hoskote. We can have the poster here, who will be coming next Tuesday to talk to you again at 7 p.m. live on the same channels on Facebook, YouTube, and Instagram. But as I said before that, once again reminding you, day after tomorrow, Thursday at 9 p.m., we are traveling to Luxembourg with a very famous poet and writer, Anna Leader, and it is part of the sideline. It is on the sidelines of the historic meeting that our Prime Minister is going to have with the Prime Minister of Luxembourg, the first time in 20 years. And this is going to happen on the sidelines on our platform, that we will have one of the very famous young authors of Luxembourg who will be taking us and, giving us a perspective to understand the country of Luxembourg culturally. So thank you very much. Thank you, Professor Dash. And I hope all of you will be back for our shows. Thank you.
In education that we are trying to promote reading. As statistics say, if 50% children in grade 5 cannot read the textbook of grade 2, then what is really happening? That's because the basic skills of reading and writing is not there. So we thought that if we have to change this cynicism, then we need to demonstrate what is possible with small contributions. So that is the idea basically that Bakul is trying to demonstrate, what is possible entirely with volunteerism, entirely with small little contributions. We asked people to contribute books to set up a public library in Bhubaneswar, and a children's library was set up in Bhubaneswar, and now that children's library has become a general library, a public library with more than 20,000 books. So that is one of the major things. Another major area of intervention is environment. A lot of environmental education, but through films and through creative methods. So we have a traveling film festival on environment. We go to schools, colleges, apartments and screen films on environment, primarily on climate change and on issues of water and sanitation, two issues that we're working in. And at the libraries also, we conduct a lot of workshops on creativity. We all want the world to be a better place, and we have our own ideas of how it can be better. The thing is, can we all start in some small way towards addressing that? Can we say that in some small way, this world is the way I want it to be because I have done something about it?